Good evening, everyone, and welcome to a week 5 of live session for basics of crop breeding and plant biotechnology. I hope my voice is audible and my screen is visible. And I guess everyone has joined by now. So we can start the session. I am Prashasti Pandey, a PhD scholar and PMRF fellow from Department of Botany, Banaras Hindu University. And if you have attended previous live sessions, in this sessions, we'll be going through some questions of previous year's assignments. I'll give you some time to solve them. If you know the answer, you can either speak it out loud or write it in the chat box, and then we can try solving it. Um in a hope that this will help you solve this year's assignment for this course. Okay, so let's begin with this session. Okay. So first question is, which of the following is not a factor responsible for inbreeding depression? Your options are increased homozygosity, expression of deleterious alleles, increased genetic diversity, and loss of heterozygosity. If you know the answer, you can tell it. Okay. So let's solve this question. So let's first understand what inbreeding depression is. Basically, inbreeding depression is basically decline in biological performance, and it is usually caused by increased homozygosity due to inbreeding. Okay. Okay, so it is usually caused by inbreeding. Now, in case of inbreeding depression, what exactly happens is Because there is continuous homozygosity, there are chances that harmful recessive alleles come into play. Okay, the alleles which are not currently being expressed because they have a dominant allele and it is overshadowing the recessive allele, which might not be uh, very useful, or rather it might be harmful for the organism, that is currently suppressed because there is a dominant gene present. But because of this inbreeding depression and homozygosity, what happens is there are chances that in some organism, that dominant allele which was protecting from the harmful effect of the recessive allele disappears, and there is now homozygosity of the recessive alleles, which is harmful. So, in case of inbreeding depression, that is possible. Then, second, heterozygosity is lost because with constantly inbreeding, there are chances that the one of the offsprings will not have a heterozygous combination and it will become homozygous. And third will be reduced hybrid vigor, which is also called as heterosis. And now this is also an outcome of in breeding depression, that because the best variety is not getting selected, because as a principle of adaptation and as a principle of increased genetic variability, there are more chances of getting a better quality offspring. But since both all these things are not present, therefore the outcome will be that the progeny will be less vigor, or the hybrid vigor will be reduced in form. So all these are outcomes of inbreeding depression. Now coming back to the question, which of the following was not responsible? So option A was increased homozygosity. So yes, increased homozygosity is responsible for inbreeding depression. It is one of the main causes. So because when inbreeding increases, your homozygosity will increase, and this will expose to recessive harmful harmful recessive alleles, and it is one of the, as I told you, that it is one of the major causes of inbreeding depression. The second option was expression of deleterious alleles. So yes, it is responsible, because when in this case, when harmful recessive alleles become more present, because in case of homozygosity there is no dominant allele which is there to mask the effect of recessive allele, so there are chances that harmful recessive alleles will express in homozygous condition, and this will basically lead to less vigor or reduced fertility and reduced survival. So this option is also incorrect, that because it is responsible. Option C was increased genetic diversity. So this is not responsible, because if the genetic variability increases, then basically the concept of increased homozygosity will actually get reduced. Because when genetic diversity is increasing, your heterozygosity is also increasing. And when heterozygosity is increasing, there will be nothing called inbreeding depression, because it is not continuously inbreeding itself. So no, genetic diversity will actually not increase the inbreeding depression, but it will actually reduce it. And this will also improve the vigor. It will not cause depression. Okay, so this is the false answer. And fourth was loss of heterozygosity. So of course it is responsible, because when heterozygosity reduces, it will increase the homozygosity, and it will cause more reduction in hybrid vigor. Okay, so the correct option is option C, that increased genetic diversity is not a factor responsible for increased for inbreeding depression. Okay, I hope this was clear. Let's move on to our next question. In the context of Hardy-Weinberg equilibrium, which factor disrupts equilibrium? The options are random mating, mutation, no selection pressure, and large population size. Okay. So someone has answered mutations. Let's see this. So basically, Hardy-Weinberg equilibrium is a condition in which allele and genotype frequencies they will remain constant in a population under ideal conditions. Okay, so Hardy-Weinberg equilibrium basically states that in a population during ideal conditions, the alleles and the genotype frequencies will always remain constant. Now, let's understand this Hardy-Weinberg equation first. So, suppose there is one allele which is capital A and the other allele is capital B, small a. Okay, so this should can we can denote this by P and the other one by Q. So now, according to Hardy-Weinberg principle, because it says that it will remain constant, therefore P plus Q will be equal to 1. Okay, so the presence of capital A and small a will always be equal to 1, and therefore the genotype frequencies become p square plus 2pq plus q square. Okay, and this will be equal to 1. Now here p square is the frequency of capital A allele, q square will be the frequency of small a allele, and 2pq is the frequency of heterozygous combinations, which is capital A and small a. Okay, so this is basically Hardy-Weinberg principle. Now, there is, you can take an example like if P value, which is frequency of one allele, is zero point 0.6 and the frequency of another allele is zero point 0.4. So, according to Hardy-Weinberg principle, P plus Q will be equal to 1, which is true because zero point 0.6 plus zero point 0.4 is equal to 1. And now, if we take as P square is equal to, will be double of zero point 0.6, which is equal to 0.36. Q square will be double of zero point 0.4, which is 0.16, and 2pq will be multiplication of this, which will be 0.48. Now, if we sum up everything, the sum will come up to 1. Okay, so this is Hardy-Weinberg principle. Now, according to Hardy-Weinberg principle, there are certain assumptions or rather conditions that are required for this whole thing to be correct. One of them is random mating, that the population that we are selecting, the condition is that the population is randomly mating. There are no mutations, no natural selection happening. The population size is large and there is no migration, which is also called as gene flow. So, if a population is basically fulfilling all these factors, then it can be assumed by assuming all these things that a population is fulfilling, then in that case, only Hardy-Weinberg equilibrium works. Now, there are certain factors which can basically disturb this equilibrium, like mutation. So, if mutation happens, then in that case, the allele frequency will not remain the same and it will not follow the equilibrium. Second is selection. In selection, there are chances that one of the three allele combinations will be given more preference, and that is why their frequency will increase because of selection. That is the reason that why basically selection will disturb the equilibrium. Then there is migration. So when a chunk of population basically moves from one location to another location, the alleles that are present in that chunk will basically get subtracted from all the population, and that it does not mean that it will maintain the equilibrium because we don't know what kind of alleles are basically moving out. So migration is also one of the reasons that can disturb the equilibrium. The another reason is genetic drift, and then there can be non-random mating. All these things can basically disrupt the equilibrium because in most of the cases either the preference is given to one of the three alleles, or the most of the populations are moving away due to geographical reasons, due to multiple reasons. They are basically shifting, or they are not allowing the increase of heterozygosity, and therefore this equilibrium gets disturbed. Now, about our question. So, which of the following, the question was talking about that which of the following is disrupting the equilibrium. So, first was random mating. So, no, random mating cannot disrupt the equilibrium because it is actually required for the equilibrium to be maintained. Second was mutation. Yes, mutation will basically disrupt the Hardy-Weinberg equilibrium because it will introduce new alleles, it will change the allele frequency, and that is why the equilibrium will get disturbed. C was no selection pressure. No, no selection pressure will actually be better to maintain the equilibrium because there is no preference given to any one allele. So this is also wrong. And fourth one is large population size. No, it will not disrupt the equilibrium. Rather, one of the assumptions of this equilibrium is the population size should be large, and it will also prevent the genetic drift, and it will help in maintaining the equilibrium. So in this question, Hardy-Weinberg equilibrium, mutation will basically disrupt the equilibrium. So option B, yes, option B was correct. Let's move on to next question. Which of the following will be a homozygous and homogeneous population? The options are hybrid made in cross-pollinated crop, hybrid made in self-pollinated crop, mass-selected variety in cross-pollinated crops, and pure line in self-pollinated crops. Okay, so two students have answered D, pure line and self-pollinated crops. So let's look at this. First, let's understand the four terms that was given in the options. First term was homozygous. Okay, so basically, homozygous is a condition in which an individual has two identical alleles. Okay, for a particular gene. So, like capital A, or small a is basically homozygous conditions. Okay, so both alleles are same in homozygous. Second one was homogeneous. So homogeneous word, so basically allele is for a single individual. Okay, but homogeneous talks about a population. So a population in which all individuals are genetically identical is basically a homogeneous population. So there will be no genetic variation within a population. Just keep in mind that the two terms homogeneous and homozygous. Okay, so homozygous will always talk about individual. Okay, and genius will talk about population. Okay, this is the important difference between homozygous, homogeneous, heterozygous, and heterogeneous. Okay, because genus is talking about the entire population and zygous is talking about one population. So okay, so this was basically homozygous and homogeneous. So that was the meaning of the two terms. Now about the options. So hybrid is made in cross-pollinated crop. So basically this is wrong because hybrids are heterozygous. Okay, so even though they are heterozygous, in case of homo, they are not homozygous. Okay, so hybrids are usually heterozygous, so they are not made in cross-pollinated crops. Option B was hybrid made in self-pollinated crops. So yes, the thing is that hybrids are also heterozygous and they are not homozygous. So again, this option is wrong. Option C was mass-selected varieties in cross-pollinated crops. So in this case, the population is heterogeneous. Okay, and it contains both homozygous and heterozygous individuals. So this is also wrong. Option D was pure line in self-pollinated crops. So this is the correct answer because in case of pure line and self-pollinated crops, both because the crops are developed by continuous repeated selfing. So in this case, all the individuals are homozygous as well as homogeneous. Okay, because there are no chance because continuously being produced by a selfing, and that too of a self-pollinated crop, the individuals that are produced will have same type of allele, and all the individuals will have same type of allele, therefore the population will also be homogeneous. So because yes, the plants that are produced are genetically identical. So in this question, that which of the following will be homozygous and homogeneous as well? So the correct answer is D, pure line in pollinated crops. And other three options, both homozygous and homogeneous together is not possible. Okay, so I hope this was clear. Moving on to our next question. The loss of fitness due to mating between closely related individuals is termed as. Options are genetic drift, inbreeding depression, hybrid vigor, and founder's effect. It's a very easy question. Okay. So we have answer as B. Someone has replied B. So yes, let's talk about it. It is actually right answer. It's very easy. We just talked about it. That, but let's understand all the four terms. One was genetic drift. Okay. So what genetic drift is it is basically random change in allele frequencies in a population due to some chance events. Okay, especially in small populations. So it basically occurs by chance. So it is not an outcome of selection. It the effect of genetic drift is the effect of genetic drift is basically stronger in small populations, and it can lead to either loss or fixation of alleles. So this is called as genetic drift. Second was inbreeding depression. We just talked about it. That inbreeding depression is basically reduced vigor, fitness, and fertility. And this will happen due to mating between closely related individuals, and it will result in increase in homozygosity and sometimes expression of harmful recessive alleles. Okay, it is usually possible in cross pollinate it. So it basically causes reduced yield and vigor. Okay. The third term was hybrid vigor, which was also called as heterosis. So, hybrid vigor is the superior performance of hybrid offspring as compared to their parents. Okay, in terms of it could be in terms of growth, yield, vigor, or resistance. So, in this case, the offspring will perform better than their parents, and it is exact opposite of inbreeding depression, and it is due to increased heterozygosity, and it is common in crops like maize. The fourth term was founder effect. So basically, founder effect is the change in allele frequency when a new population is established by smaller number of individuals from a large population. So basically, it is an example of genetic drift. Okay, like there is a huge population and small chunk of this population basically moves out, and then this population, because it is established by small number of individuals from large population, the change in allele frequency is called as founder effect. So in this case, it is a type of genetic drift, and new population. This population will basically have reduced genetic variation. Okay, and allele frequencies may differ from the original population, because in this population we don't know what kind of alleles have moved here. So that is why the frequency might not follow the same equilibrium or the same ratio as it was there in the original population. Okay, so this effect is called as founder effect. Now, according to the question, the correct answer was in breeding depression, that it is the loss of fitness, vigor, and fertility, basically caused by mating between two closely related individuals, and happens because of increased homozygosity. In this case, harmful recessive alleles will become expressed, and it will lead to reduced survival, growth, and reproduction. Why other options were wrong? Genetic drift was wrong because random change in allele frequencies. It is not specifically due to inbreeding. Second, C option was hybrid vigor. So it is exact opposite of inbreeding depression. So it will basically increase the vigor and fitness and not reduce it. And founder effect will only happen when small group will form a new population with limited genetic variation. So according to the question, loss of fitness due to mating between closely related individuals is termed as inbreeding depression. Okay. So yes, option B was correct. Now let's move on to next question. The total genetic information carried by all members of population is called as genetic drift, genetic flow, genetic pool, and genetic load. Okay. So let's solve this question. So, in previous one of the previous questions, we have already talked about what genetic drift is. That it is a random change in allele frequencies in population due to chance event, and especially in small populations, and it can occur by chance, not due to selection. And the strong effects are in small population, and it may lead to either fixation of or loss of alleles. Now, about gene flow. Okay. So about gene flow is basically transfer of alleles or genes from one population to another population through migration of individuals, or in case of plants, it is migration of gametes or like pollen grains or seeds. Okay. So that is called as gene flow. Now, gene flow will basically introduce new alleles into population because there are chances that either the individuals or the gametes are coming from another population which was not present in the new population. So there are chances that new alleles will get introduced. There is increased genetic variation because of gene flow, and it will reduce the genetic differences between two populations. Okay, so like if there is one population and there is another population, if there is a transfer of alleles from this population to this population, there in this population the genetic diversity will increase, but amongst these two populations the genetic differences will start reducing because eventually these alleles will start increasing here also, so the genetic differences in the two populations will reduce. Okay. Third was gene pool. Gene pool is basically total collection of all the genes and their alleles that are present in individuals of a population at any given time. Okay, so gene pool will basically represent the total genetic diversity of a population, and larger the gene pool, higher the variation. Okay, also because gene pool increases, larger gene pool will have more variation, therefore, it will also increase the adaptability and eventually evolution. The fourth term was genetic load. So, genetic load is basically presence or accumulation of deleterious alleles in a population that is eventually going to reduce its average fitness. Okay, so genetic load will basically it is caused by harmful mutations, and the higher chances of genetic load to increase may be due to inbreeding, and because the more number of harmful mutations are present in that population, their survival and reproductive ability will get reduced. Okay, so this is called as genetic load. Now, coming back to our question, the question was that total genetic information is carried by all the members and it is called as. So the correct answer is gene pool, because gene pool's definition is total genetic information which is present in all the populations, all the individuals of a population at any given time point. We have already talked about this, that it includes all alleles of all genes. It represents the genetic diversity of that population, and larger the gene pool, greater will be the genetic variation and better will be the adaptability of that particular population. Why other options are wrong? Genetic drift talks about the frequency changes that too by chance, so it is not the total population, total allele representation. Gene flow is basically the movement of genes between two populations through migration, so no. And fourth was genetic load. It is basically increasing of harmful or deleterious alleles in a population, so no. That is also not talking about the entire information. So option C is correct in this case. That total genetic information by all members of a population is called as genetic gene pool. Okay. Okay. So our next question is, which crop is least likely to exhibit inbreeding depression? Okay, so let's solve this question. Okay, so inbreeding depression is least in self-pollinated crops because for obvious reason they are highly homozygous due to conditions like selfing and selfing over many generations. Okay, so wheat is the correct answer here. The reason is wheat is a self-pollinated crop and it is naturally homozygous and it will show very little or no inbreeding depression. Correct. Then soybean. Soybean is also self-pollinated crop, but it will show low inbreeding depression. But wheat is most classic example. Sugar beet is basically cross-pollinated crop, so it will have severe inbreeding depression, and same is the case with carrot. So in this case, wheat is the crop which will exhibit it is very least likely to exhibit inbreeding depression. Okay. So the next question is the frequency of two alleles of a gene in population are 0.10 and 0.50. If the population is an Hardy-Weinberg principle, what will be the frequency of heterozygous individuals in the population? The options are 14%, 20%, 32%, and 10%. So this is a numerical question and it's interesting one, so you can try solving it. Okay, so let's solve this question. So the question basically works on the Hardy-Weinberg equilibrium. So we have talked about that in Hardy-Weinberg equilibrium, P plus Q equals to 1 and the heterozygous frequency is 2pq. Okay. So now according to this, the in the question it says that capital A frequency was 0.1, which can be denoted by P, and Q is small a frequency, which was 0.5. Okay. So now according to Hardy-Weinberg principle, P plus Q should be equal to 1, which is not happening right now. Okay, it is 0.6 right now. So, because they do not sum up like this, we interpret that we also need values that there are some heterozygotes present in this particular population. Okay, so now to calculate the heterozygous populations, we need 2pq value. So 2pq value will be 2 into 0.1 into 0.5. Okay, so this answer is 2 um, 2 into 0.05, which is equal to 0.10. So now converting this into percentage, it will be multiplied by 100, so which will be equal to 10%. Okay, so the answer in this question is 10%. That if what will be the percentage of heterozygotes in the population? Answer is 10% will be heterozygotes, because your P was 0.1 and the Q value was 0.5. Okay. So now calculating by the previous formula that we just talked about, the answer is 10%. Okay. So the next question is also very similar. The frequency of dominant homozygotes in a diploid population is 0.6. Assuming that population is in Hardy-Weinberg principle, the frequencies of two alleles are. Give it an attempt. Has anybody got the answer? Okay. So we'll try solving it. So again, the Hardy-Weinberg principle of P square plus 2pq plus q square is 1, where your P square will be of one allele, q square will be of another allele, and your 2pq will be basically heterozygous population. Okay. Now coming back to the question, the P square value was given as 0.64, and so because P square is 0.64, so in that case, square rooting the to get the P value, square root of 0.64 is basically 0.8. Now, because we know P, we can calculate Q from it, because P plus Q equals to 1. So now because we know P value, so 0.8 plus Q equals to 1. So Q value will be 1 minus 0.8. Here Q is 0.2. Okay, so this was the question that we were supposed to calculate the values of P and Q, which is of dominant allele and recessive allele. Dominant allele is represented by P and recessive allele is represented by Q. And now we know the values. Value for P is 0.8 and value for Q is 0.2. Okay, so this is the correct answer. So according to this, your option A is correct, 0.2 and 0.8. Okay, this will be the frequencies of two alleles. Okay, so now moving on to next question. Which hypothesis of heterosis suggests that heterozygous individuals have superior traits compared to both homozygous parents? The options are dominant hypothesis, over-dominant hypothesis, mutation hypothesis, and inbreeding hypothesis. Okay, so let's talk about this. In this question, there are four terms. We'll just understand those terms first. First was dominance hypothesis. So dominance hypothesis basically states that heterosis will occur because of dominant alleles, and it will mask the harmful effect of recessive or deleterious alleles. Okay, so in this case, basically, harmful recessive allele, because if there are two alleles present and this allele is harmful, in case of heterozygous population, effect of this allele is masked. So in hybrid cases, it becomes superior because the harmful alleles are not usually expressed. So the example is like, if the parent has capital A, small b, and the other parent has the opposite combination, so the hybrid will be capital A, small a, and capital B, small b, and this will be of superior type because in this case, the harmful effect of both the recessive alleles are masked by their positive alternatives. Okay, so in this case, dominant alleles will basically hide the harmful effect of recessive alleles, and this is called as dominance hypothesis. Second is over dominance hypothesis. So over dominance hypothesis will state that basically heterozygous genotype is superior to both homozygous parents. That is, your capital A, and small a, and heterozygous will be superior than both the homozygous genotypes. So in this case, heterozygous has better performance than parents, and superiority is due to heterozygosity itself. So again, the example is capital A, and small a will be of lower performance as compared to capital A, small a, which will be of higher performance because there is increased heterozygosity. So this is called as over dominance hypothesis. C is mutation hypothesis. So, as the name suggests, that mutation hypothesis will state that heterosis will occur due to accumulation of favorable mutations and elimination or masking of harmful mutations. So, in this case, basically, mutation is occurring and it is occurring through genetic variation. But only favorable mutations will be kept, and that will help in improving the hybrid performance. So, mutation is basically contributing towards genetic improvement, and therefore its variation, and thus the outcome of that mutation will be better performing than its parents, which is heterosis. And D type is inbreeding hypothesis. So inbreeding hypothesis explains that loss of vigor will occur due to inbreeding, and heterosis is restored vigor by increasing heterozygosity, because in case of inbreeding, your homozygosity is basically increased. Okay, so when homozygosity is increased, so in this case what happens is that our basically uh, viability or vigor is basically reduced in this case. So when due to inbreeding only the vigor is restored or the heterozygosity is restored, then in that case it is called as inbreeding hypothesis. Inbreeding will basically increase so this I explained, that inbreeding will increase homozygosity and it will reduce vigor, but hybridization will increase the heterozygosity and it will restore the vigor. So hybrid vigor is exact opposite of inbreeding depression. Now coming back to our question, the correct answer is over dominance hypothesis because it states that heterozygous genotype is superior to both homozygous parents. Okay, in terms of vigor, in terms of yield, fitness, and other desirable traits that we have already learned by now that what over dominance hypothesis is. Okay, so now why other options are incorrect? So dominance hypothesis, because the question was talking about that which hypothesis suggests that heterozygous individuals are superior than homozygous parents. So in case of dominance hypothesis, basically dominant allele is masking the effect of recessive allele. Okay, so it is not talking about the parents being homozygous. And heterozygous is not necessarily superior to both homozygous parents. The parents were not homozygous. And in case of mutation hypothesis, it is not related to this because it is caused by mutation. And inbreeding hypothesis, it explains about inbreeding depression and not heterosis. So option B is correct, that over dominant hypothesis is basically in case of heterosis, it will suggest that heterozygous individuals have superior traits compared to both their homozygous parents. Okay. Now coming back to our last question. In a population of 5,000 individuals of plant species, genetic variation at a single locus leads to different fruit shapes. The trait exhibits incomplete dominance for two alleles. The population consists of 1500 individuals with genotype QQ, 2000 individuals with genotype capital Q small q, and remaining 1500 individuals with genotype capital Q capital Q. What is the frequency of Q allele in the population? Okay, let's solve this question. So now we have to find the frequency of allele Q. Okay. So now to find this, we will have to use the formula, which is 2, because we are talking about, because the question is stating specifically about incomplete dominance. Okay. So in this case, the formula which will be used is this. that q is equals to 2 times small q plus capital Q small q upon 2n, where your small q in question, your small q, it is written as 1500 individuals. Okay. And for capital Q small q, it is 2000. Okay. The total population in the question, it is given as 5000 in a population of 5000 individuals. Okay. So n is 5000. So 2n will be 2 into 5000. So which is equal to 10,000. Okay. So 2n is 10,000. Now just put all the values in this. So each q individual will contribute to 2 alleles, right? Because it has two small q's. So that means that for allele q, it will be 1500 into 2, which will be equal to 3000. Okay. Now in this case, when capital Q and small q is there, so q is only contributing once, right? So 2000 into 1 will be 2000. Now total Q alleles will be summation of this two, which will be equal to 5000, right? So now the step 2 will be to calculate the frequency of Q allele. Now it is very easy to calculate the frequency. It will be basically 5000 upon your 2n is the population number that was given for the formula, and 2n was 10,000. So if we calculate this simply, it is 0.5. Okay. So this is the answer, 0.5. Frequency of Q allele in the population is 0.5. Okay. So I hope this was understandable. And if there are no doubts, we can end this session and we will meet again next Tuesday, same time.